So today we're here to listen to Chelsea's thesis defense, where she's going to be talking about a bunch of research she's been compiling after a really cool field campaign last summer, focusing on carbon and nitrogen cycling and how that's intertwined with microbiology. She dove headlong into the lab when she joined the lab 2 years ago, after several years in industry and before that an undergraduate at Cornell. And while she'll just be talking primarily about the work she did as related to understanding carbon and nitrogen cycling, she's also been involved in several other projects in the lab, understanding sort of the differences between live extracted microorganisms through RNA versus DNA, better understanding some other elements of biogeochemical cycling in the field, as well as some help with sulfate-reducing bioreactors. But that's a topic of conversation. So with that, I'll turn things over to Chelsea. She has the floor, and thanks for coming.
All right, thank you guys. This is a great turnout. Josh, thanks for that great introduction. So I've been working on this project about pine beetle's effect on soil nutrient cycling for the last year and a half. And this is actually a picture from the field site where my colleague Brent and I worked last summer and collected all this data. I think most people here in Colorado are pretty aware of the mountain pine beetle epidemic that's going on for the past decade or so. It's hit Colorado particularly hard because we have a lot of lodgepole pines. But it's not just a local phenomenon, it's happening all over the northern hemisphere, primarily in temperate forests. This shows the extent of the mountain pine beetle infestation in North America on the West Coast, but there's hundreds of species of bark beetles that all infest different trees and are causing similar issues in the eastern US and also in European forests. And so one of the reasons, pine beetles are endemic to this area, and there have been past observations of outbreaks, but the current outbreak has been much more severe and much more intense, and this has been linked to climate change. So with warmer summers and milder winters, pine beetles are able to maintain higher population levels, and trees are also stressed from climate-related changes such as drought. So this isn't just affecting the trees, though, it's also affecting the soil, the water, and the atmosphere around the trees. One example of how this affects water quality has been done by my colleague Kristen Mickelson, and she showed in a Nature climate change paper that organic carbon is actually increased in watersheds that have been infested by pine beetles. And downstream at the water treatment plant, this causes issues because it increases the potential for toxic byproducts after the chlorination process. This is also affecting climate change. So we already talked about how the bark beetle infestation has been exacerbated by climate change. Well, it's also likely that bark beetles are going to have a positive feedback and also exacerbate climate change through the release of greenhouse gases, and since the trees are no longer taking up carbon dioxide. And so underlying these big picture issues is really just soil, carbon, and nitrogen cycling. And this is the framework for the work I'm going to present for you. And basically just to get you thinking about the way I've been thinking for the past year and a half. A living, healthy tree is going to be taking up carbon dioxide and converting that into organic carbon, which is food and energy for itself. It's also depositing some of that food through its roots to subsurface microorganisms that help it live. And then you also have the tree producing litter, and that starts the top-down decay processes and feeds into these nutrient pools. And one key concept is that microorganisms are responsible for regulating these pathways and shuttling these nutrients from pool to pool. What happens after bark beetles come in and infest a tree? Well, they burrow under the bark, eggs, and they also deposit blue stain fungus. It's a symbiotic fungi that all bark beetles have, and it creates that characteristic blue staining that most of us have seen on lodgepole pines. And so this prevents the tree from transporting water and nutrients throughout the system. So after bark beetle-induced death, this changes the inputs to the system. The trees stop taking up carbon dioxide, and they also are no longer converting that into food and depositing it into the subsurface. So those microorganisms lose the source of food. And then something that's unique to bark beetle-induced death is that you get a pulse of litter. So 3 to 5 years after a tree has been infested, it enters the gray phase, as it's known. And it will drop all of its needles over the course of 1 season, which is pretty rapid. And interestingly, this litter is also nitrogen enriched, more so than naturally aged litter. So this begs the question, how are these changing inputs affecting the downstream nutrient pools, or how is nitrogen cycling changed? And this is not a new question. Scientists have been working on this for decades, ever since we started becoming interested in bark beetles. But what's come out of that body of work is that there's a lot of variability. It's hard to predict downstream effects. And so the new question becomes, what's the source of this variability? And how can we understand that so that we can actually make predictions about what will happen to water quality and climate change? And so there's been some evidence lately that shows that one of the things we might be overlooking is the number of trees that have been killed in a watershed after beetles have infested. Some of this work actually comes again from my colleague Kristen Mickelson and Brent Brouillard as well, where they show that the organic carbon entering the water at water treatment plants was really only increased in the watersheds that they looked at with high levels of tree mortality, but not in the watersheds with low levels of tree mortality. And we've also seen this at the tree stand. We compared 2 analogous sites and saw changes to soil parameters, but only in the site with high levels of tree mortality and not in the analogous site with low levels of tree mortality. And furthermore, we've also seen this in other fields that deal with forest disturbance, such as logging. So in logging literature, you can see that there's actually a threshold of trees that need to be removed before they start seeing an accumulation of nitrogen in the soil. And so this led us to develop this hypothesis that there's a threshold of tree mortality before you can see downstream changes in the nitrogen cycling and the microorganisms that do this. So my colleague Brent and I went out last summer and we developed a field study to test this question. And so we started out by selecting our field site and identifying sample trees. Our field site is Officers Gulch. It's right off of I-70, about an hour and a half west of Golden. And this is an aerial photo of the site from 2006, pre-infestation. And then sometime before 2010, pine beetles came in, and it might be hard to see, but you can see some trees in the red phase and some in the gray phase. So the next thing we had to do was quantify the surrounding tree mortality at our sample trees. So we selected 7 green healthy trees as our controls, and then looked at 31 gray phase trees that were surrounded by varying extents of tree mortality. And to get that number, we designated an 8-meter radius, and then we counted the percent of dead trees within that radius. And the reason we ended up at 8 meters is because lodgepole pine root system typically extends about 4 meters from the trunk. And so by doubling that, we wanted to include any trees that might have interactions with our central sampling tree. And so here's some pictures of our field site when we were out counting trees. And lodgepole pine forests are pretty interesting. They tend to grow fairly densely packed, very monoculture, so there's not a lot of other species there. And one important point I want to make also is that these environments are nearly always nitrogen limited. So we'll come back to that later. So after we measured our surrounding tree mortality, we went out and collected the soil samples. At our site, we had very distinct, well-developed soil horizons, so we sampled from all three. And that just means that there was a clear visual demarcation between each soil layer. So the top is the litter, the center one is the organic horizon, and the bottom is the mineral. And that horizon extends down to bedrock. So we analyzed our samples down two pathways. The first one we'll talk about is the geochemistry analysis, and then the second one is the microbial community analysis. So to analyze the geochemistry, we quantified all of those carbon and nitrogen cycling pools that I showed you on the conceptual framework slide. For carbon, that's dissolved organic carbon. And then we also use specific UV absorbance or SUVA, to quantify how recalcitrant that carbon fraction is. It's a measure of the aromaticity of the carbon molecules, and aromatic ring structures have stronger bonds and so are more difficult to break, making them more recalcitrant. And then for nitrogen pools, we measured total dissolved nitrogen. And the total nitrogen pool has three components in forest soils. That's ammonium, nitrate, and organic nitrogen. For ammonium and nitrate, we measured that using spectrophotometer methodology. And in organic nitrogen, there aren't good analytical techniques for quantifying that. So it's typical in this field to simply subtract ammonium and nitrate from total nitrogen to get your organic. And then a really key parameter is carbon-nitrogen ratio. So that's the intersection of these two nutrient cycles, which are highly interdependent. And so you can think of the carbon to nitrogen ratio as the relative abundance of carbon and nitrogen in the substrate that the microorganisms feed on, used for food and energy. So there are three key results in the geochemical analysis. And the first is that there was a clear ammonium threshold in the upper soil horizons. So we have ammonium on the y-axis and the surrounding tree mortality on the x. And if we look at the litter and organic horizon first, you can see that ammonium only starts to accumulate after approximately 40% tree mortality. And then in the mineral horizon, it's not dependent, or it's not related to the extent of tree mortality, but most gray phase trees have higher levels of ammonium. And so from this, there's some interesting conclusions we can make. The first is that ammonium is the most bioavailable form of nitrogen. It's the ideal form for organisms to take up. And when you see it accumulating in an environment, it by definition means that the environment is nitrogen saturated. And so remember, as we talked about earlier, lodgepole pine forests tend to be nitrogen limited. And you can see if we look at our green phase control trees that nitrogen makes up less than 10% of the total nitrogen pool. And then the second take-home point on this is actually an idea that I forgot to mention, but so inorganic forms of nitrogen and carbon are much more mobile, which means that they can exit the system more faster time frame than the organic forms. So if you take ammonium or nitrate or any of the gases, they're more mobile than the organic nitrogen or organic carbon. And so what we're seeing is that in undergrade trees that are surrounded by high levels of tree mortality, you're transitioning to a state where there's more mobile forms of nitrogen. So this is really cool, but we also want to know why this is happening so that we can extrapolate to other sites and understand the mechanisms. And if we look at the carbon to nitrogen ratio relative to tree mortality, there's a similar pattern. as with the ammonium response. Kind of inverse though. So in the litter and organic horizon, carbon to nitrogen ratio decreases as the extent of tree mortality increases. And then in the mineral horizon, again, carbon to nitrogen ratio is lower regardless of the number of trees that are dead in the area. And so this supports our conclusion that the system is going to nitrogen saturated because nitrogen is increasing in abundance relative to carbon. And this also makes sense if we think about how the inputs have changed, right? So if we think back to that conceptual framework slide, we know that after trees die, they're going to stop depositing carbon substrates, that food for the microorganisms. So we lose a source of carbon in the subsurface. And then we also know that there is nitrogen-enriched litter that is dropped. And so there's an increase in nitrogen. So how does this translate to the altered nitrogen cycling that we're seeing? And so this slide is the concentration of ammonium versus the relative abundance of carbon and nitrogen. And so I want to make an analogy. I think anybody that's involved in, who here is involved in composting or any gardening? There's a couple people. Great. This is going to make a lot of sense to you guys. So for composting, it's the carbon to nitrogen ratio is a key parameter. And you know that if you go too high on the carbon nitrogen ratio end, that you will kind of slow, you won't get optimal decomposition. And you also know that if you go too low on the carbon and nitrogen end, that you'll get kind of a smelly decaying organic matter. And that smell is actually ammonia. In ammonia and ammonium are analogous. In forest soils, it doesn't volatilize because it's at a low pH. So basically what we're seeing is that as the relative abundance of carbon and nitrogen decreases, there's a certain tipping point, and at this point nitrogen becomes in excess, and there's an accumulation of ammonia. This likely is occurring because since there's excess nitrogen, the microorganisms in the system need to waste it rather than incorporate it into their bodies. So just to recap, we're seeing that the extent of tree mortality in an area is associated with a switch from nitrogen limited to nitrogen saturated environment. And ammonium is accumulating when nitrogen is in excess, and this is likely because of the microorganisms wasting ammonium. And finally, the total nitrogen pool is more mobile under trees with increased levels of tree mortality. And if you have a more mobile nitrogen fraction, this can have downstream effects on water quality and climate change. So let's go back to our second pathway of analysis and take a look at what's going on with the microorganism community. So we know that there's a changing nutrient environment that they need to adapt to. And microorganisms can adapt in two primary ways. The first is that they can change who is there. So this means changing the microorganism, the community members in that in the soil. And so this is like your typical natural selection scenario where there's competitive advantages changing and you see an increase or decrease in the community members that make up the total microbial group. The second way they can adapt is by changing what they do. So we already talked about this first one, which is saving or throwing away nutrients, right? So when there's excess nitrogen, instead of assimilating nitrogen into their bodies, they waste it as ammonia. And the second one is enzyme activity. So enzymes break down their food and make it accessible so that they can convert it into biomolecules. And so when nutrients change, certain molecules may become more or less valuable. And so by changing enzyme activities and production, they can adapt to that environment. And so one analogy for this is how a business adapts to changing economic environments. So if for example, in the recession that we just went through, what does a business typically do to adapt to that? Yes, I'm asking you guys. Oh yes, they fire people. Great, thanks, Kristen. And so that would be an example of changing who is there. Another way they could adapt is by buying more efficient machinery or producing a different product that people like more. And so those are examples of changing what they do or their activities. So let's start with who is there, and then after we'll go into how they change their activity. So the methods for this is to do 16S rRNA gene sequencing on an Illumina platform. And so basically all you do for this is extract DNA from your soil sample, and then you copy a small section of that, which is the rRNA gene. John, I've always assumed that this is the rRNA gene. Accurate? Yeah. And so then you sequence it. So you get a list of what nucleic acids are part of that sequence. And we can compare this sequence to a database which identifies what I'll very loosely call species. I apologize to the microbiologist in the room. And then you can also get an idea of the abundance of those microorganisms. So there's 3 key results from this. The first is that bacterial diversity increased under gray phase trees. And this has been shown through some of Kristen's work as well, and in a paper I worked on with her. And then the second thing we can do is we can look at the overall community structure. So this is a principal coordinate analysis plot, and it's a way to take a multi-dimensional data set. So in this case, it's in one sample, it's the list of an abundance of all the species in it, and compress it into a 2D format that we can visualize. And so you can think of these as distances. So the further away another sample is from this one, the more different it is in terms of the phylogeny. And so we'll walk through so I can show you how they cluster, but all of these cluster significantly. And so the green triangles are green phase trees. The gray squares are going to be the gray phase trees that are surrounded by less than 40% tree mortality, which we define based on the ammonium threshold. And then the black circles are the high impact trees, or the gray phase trees surrounded by high levels, greater than 40% tree mortality. Okay, so for the litter layer, I've circled the green phase trees and the gray phase trees because they overlap really well. And then the high impact trees fall outside of these clusters. And so from this, we can conclude that in the litter, the green phase and low phase trees have similar bacterial communities. And in the high impact trees, the communities are different. And so this clustering changes as we increase depth. So in the organic horizon, all 3 groups cluster separately. And the low impact trees are a transitional state or intermediate state between the green and high. And then in the mineral horizon, the deepest layer, we see green phase trees cluster separately from all of the gray phase trees, regardless of how much they were impacted. And so the overall conclusion we draw from this is just that the bacterial communities are changing with after beetles infest an area, and in the upper soil horizons that it's related to the extent of impact in the nearby area. So, and finally, we can go a little deeper and we can look at specific groups of organisms where we know what they're doing and if they have a key role in the system we're looking at. And so in this case, Nitrospira is a really interesting organism. It's aerobic, which means it requires oxygen, and it's a nitrifier, which in this case means that it primarily shuttles nitrite to nitrate. It's also been shown to have a role in ammonia oxidation. And so this plot is on a log. So it's actually quite a dramatic change, but what we're seeing is that at low carbon to nitrogen ratios, there's an increase in abundance of nitrospira. And this makes a lot of sense because low carbon to nitrogen ratios means that nitrogen is more abundant, and nitrospira uses nitrogen, inorganic forms of nitrogen, as its food source. And we also know, or as its energy source. And we also know from the geochemical data that at low carbon nitrogen ratios, there's more inorganic carbon in the system. So it plays. Okay, and the second analysis for the microbial community was looking at enzyme activity. And this was a really fun assay to do. What you're looking at is in each of these wells, there is a soil sample. And then this molecule over here is our fluorescent reporter. Because of its aromatic ring structure, if you shoot a specific wavelength at it, it will absorb it and then re-emit at another wavelength. And so we can detect that reporter molecule using spectrophotometry. You can chemically bond substrates of interest to this hydroxyl group. And when the substrate is bound to the reporter molecule, it won't fluoresce, it's quenched. And so when we add that combination to these samples, we can measure how much of this reporter molecule is released using spectrophotometry. So I decided to look at 4 different substrates of interest. And these were selected because they represent increasing degrees of recalcitrance. So starch is the least recalcitrant. Humans eat it, we use it for food. And then cellulose, metabolize cellulose, but it's fiber, so it's good for us. Gotta make sure you get your fiber. And then chitin. Chitin makes up the exoskeleton of insects, so you've probably touched chitin before if you've ever played with bugs. It also is the cell wall of fungi, and interestingly, it's the only molecule here that contains nitrogen. And finally, we also looked at lignin. It's the most recalcitrant, as you can see by how complex it is, and that makes up plant cell walls. So the starch and cellulose, I didn't find any interesting trends with, so we won't talk about them. But we can look at chitin and lignin. So chitinase and lactase are the enzymes, and this shows their activity versus carbon to nitrogen ratio. And interestingly, for both enzymes, the activity decreases at low carbon to nitrogen ratio. It's difficult to explain why without actually doing experiments and testing hypotheses, but we can throw out some ideas, I guess. On one hand, it's a little bit counterintuitive because, as I said before, lignin and chitin are carbon-containing molecules, and we know that in this environment, carbon is more limited than nitrogen. And so you would think that enzymes would be, enzymes that break down these carbon molecules would be useful. But one thing that might be happening with chitin is because it's a nitrogen-containing molecule. And we know that nitrogen is in excess when we're in this region. And so maybe as a nitrogen scavenging enzyme, it's no longer as useful, and that's why activity is decreased. So those would be some interesting like future directions to study. And so just to go over these conclusions, we observed a threshold ammonium response of after 40% tree mortality in the upper soil horizons. And this was associated with a transition from a nitrogen-limited environment to nitrogen-saturated environment. And in nitrogen-saturated environments, the nitrogen fraction is more mobile, and this can have downstream impacts on water quality and climate change. And finally, the microbial community is clearly adapting to the extent of tree mortality, and it is adapting both through who is there and also what it's doing. And some future directions that this work could take is to actually test some of these ideas in the lab where we can control the variables and move these correlative relationships to causal. We did not analyze the fungal community role either, and that would be good. They're the other half of the microbial community, and most of this work focused on the bacteria. And finally, the results from this suggest that greenhouse gas flux may be a serious implication after pine beetle infestation. And so I think it was a really productive thesis. I'm co-author on 2 papers. One of them is the sulfate reducing bioreactor work that Josh mentioned, and another is work with Kristen, also on pine beetle systems. And I'm preparing this research to submit this summer. So thank you guys all for coming. I especially want to thank my committee, Dr. Josh Sharp, Kristen, and John Spear in the back. And everybody else I've listed here has either directly influenced or helped out processing samples or given advice and ideas. And of course, want to thank family and friends. So I'll take questions. Dina?
So at the beginning, you mentioned that you did some SUVA work.
Yes.
Did that direct your choices of those enzymes, or where did that come from?
So I did a report about the SUVA data, which was interesting and supports a lot of the stuff that I did report on. But in general, or not in general, but so SUVA was increased under gray phase trees. And so we hypothesized that because you have that pulse of litter, pine needles are very recalcitrant. And so we hypothesized, of course, that there would be more recalcitrance. And so that was why we looked at the spectrum of carbon recalcitrant for enzymes.
Okay. And then this is kind of more of an overarching question. It seems like in the mineral layer was a lot more resilient to the changes that you saw maybe in the organic and litter layer. So that I think that first ammonia graph you showed, for the litter and organic layer, you see like a really steep change. And for the mineral layer, you see kind of like a flat. And then also in your PCOAs,
well, so I would actually, I mean, that's a really key and subtle part of this, but I would say it's actually the opposite, and that the litter and organic layer were somewhat buffered at low levels of impact. Okay. And the mineral horizon tended to respond whether, as soon as the tree died, it didn't matter how many trees around it were impacted.
Okay. So I got it wrong.
Well, it was a good trend that you noticed, but it was just the reverse. Yeah, absolutely.
Okay, cool.
Anybody else? Yes, Tess.
I'm curious if you ended up doing any estimations of total abundance, microbial abundance, because I know that you were thinking about doing ATP and you did it.
We did, yeah. I did do ATP, and it didn't change, which is an interesting finding in and of itself, but yeah. Skylar?
I have 8 questions. Pick where you want to take it.
Great, I love those ones.
It's interesting to see a very clear correlation between C and ratio positive parameters, and I was kind of surprised by that in a good way. But have you looked at the actual magnitude of those pools and all, and do you see anything interesting with those? How is the ratio of the actual?
Yeah, we did look at that, but you have to appreciate that the carbon and nitrogen cycling, they're so intertwined that the absolute abundance of carbon doesn't really matter too much, because it only matters relative to how much nitrogen is there, because the microorganisms are using this as a substrate. So for example, if nitrogen is limiting, then you can add as much carbon as you want, and it's not going to change how the system works, and vice versa.
Do you see anything, I guess, striking though, any big variance between green and gray phase?
Yeah, I mean, we can comment on that. So there's, for example, if you just look at green and gray phase trees, there's a decrease in the amount of dissolved organic carbon. That's in the mineral horizon. And that's pretty interesting because we know that the roots are no longer depositing those simple carbon substrates. And so I think that probably reflects that. In the litter and organic horizon, there weren't very clear trends. And with nitrogen, there was a slight increase if you compared green and gray phase trees. But the really key parameter is how their abundance relative to each other. It's a good question, Zach.
In terms of like, you talked about feedback towards global warming. So overall, do you think that the pine beetle death will cause an increase in greenhouse gas emissions, or do you think maybe because eventually these forests are going to regrow, starting with younger trees and older forests that are known to take up less CO2 because they're not growing as quickly? Do you think there's going to be a net increase or decrease in greenhouse gases, let's say, over the next 20 years?
That's a great question. I mean, and that's like the question, right? That's what I mean. If somebody could definitively say that, they would, yeah, that would be like a Nature paper. But I mean, you're right. It's a dynamic and temporal thing, right? So, what the carbon and nitrogen storage balance immediately after a pine beetle infestation is going to be different than 10 years down the road. The system that we're looking at is about 10 years down the road, a little bit less than that. And so that's pre-regrowth. So there's not a lot of regrowth yet. That's coming in the next couple of years for this site. And so trees take up carbon dioxide, but we also have to think about the soil respiration. And we also have to think of other trace gases like methane and nitrous oxide, which are actually more potent greenhouse gases. And people have looked at carbon dioxide. It's probably a net neutral change. For nitrous oxide and methane, not as much research has been done. I think the work we're doing right now suggests that you're going to see an increase in nitrous oxide. And other people have shown it with tree girdling experiments. And then methane, forests are actually a net sink for methane. And this tree girdling experiment I referred to has actually shown that it's not as good at taking out methane in the atmosphere. So I think with the trace gases, you'll actually see an increased flux. And for people watching, they can ask questions too, I think. I don't know how to though.
I'm trying to look at it on my phone.
No one is saying anything?
I'm sure you can maybe
Okay.
turn on my laptop.
Do we have watching?
People are watching.
There was four. You got a little bit of online updates.
Okay. Okay, well, if anybody wants to watch or ask questions online, you can put them into that chat, I guess. Anybody else questions for me? Yes.
What happens if you plant a tree? How fast do you think these are going to respond if you put a seedling in?
I mean, that's an awesome question. I mean, so I think a seedling would love this environment. You have a ton of dead trees, so you don't have a lot of canopy blocking the sun, and you have a lot of ammonium in the soil, which is the ideal form of nitrogen for trees. So there have been people looking at succession and seeing if there's a more rapid growth right after these events. And nobody knows for sure if that's actually going to happen. One interesting paper I did read, though, is that the fungi that So the ECM, the ectomycorrhizal fungi are depleted after this comes through. And so that actually makes it more difficult for the trees to grow and move into this area. So it's mixed results again, but good question. Dina?
But like succession, I mean, what if you did grass and like shrubbery first and then these trees coming in, so it wouldn't be like, you wouldn't just skip the weed part.
I mean, all of it's happening at once. It's not quite as linear as it looks in the textbooks.
Oh.
So even at our site, there are areas where there's saplings coming in. So, yeah.
Any other questions from the room? The audience is time, and then we'll send the audience out and they get ready, so we're not gonna ask any questions. All right.
Thank you, guys.
Thanks, Chelsea.